Welcome everybody from the Health, Safety and Environment Group of RACI Victorian branch, and also welcome to Envy Smart of Melbourne University and those who've logged on through that means. So welcome everybody, and we have to start with Brad Clark of Melbourne University to present his title. It's all about polyfluoroalkyl substances, PFASs, chemicals discharged to the environment from an illicit chemical stockpile fire, which is the one that happened a while ago now in Somerville Road, West Footscray. There's an interesting article in the newspaper today about it, showing the charges that have been laid and against the company and against the director. So I think if we're all ready, let's have you go, Brad, please.
Sure thing. Well, thanks very much, Ian and Richard, for the invitation to come and speak here tonight at RACI. And I just would also like to acknowledge my team members who have contributed to this work, in particular Jay, who's also speaking later, Drew Sabathe, Madison and Tom Hennessy. Now, Tom's not from my group, but he's given me a lot of help. So I'm actually going to use this as a case study to sort of weave in some ideas around emerging contaminants and analytical chemistry. So if you had have said 100 years ago that every single person in the world would have synthetic chemicals in their bodies from the moment of conception until death, people probably would have thought it was doomsday statement and not likely to be true. But as far as we are aware, the chemical revolution has resulted in an unprecedented exposure to humanity with a range of chemicals that are accumulating in our body. And the worst of these chemicals are persistent organic pollutants, and these chemicals are accumulating in our body. And we've seen sort of generations of these chemicals, and the first ones that were really marked as being pretty bad, which were the persistent organic pollutants, or the organochlorine pesticides and PCBs. We're sort of seeing second generation pollutants coming through, and PFAS is the most famous example of that. And I'm going to hopefully try to weave in a story today of how we're really failing in our historical lessons, and are using the Footscray fire as almost a case study of how we can sort of improve to manage chemicals a little bit better as well, and how analytical chemistry actually has a really central role in helping us understand environmental pollution. And Jay's going to talk more about the health effects of exposure to PFAS, but for me, the biggest issues of exposure to these chemicals can be there's reproductive health problems, impaired immune system, and neurological damage. And I often show this picture where you see this is side-by-side villages in Mexico, and you could see very clearly cognitive damage of young children that were exposed to elevated levels of pesticides. So the risks that we are sort of playing with our health are significant. And this leads me on to PFAS, which is really this, it's probably the issue of a generation outside of climate change, and it's maintaining the entire environmental sector pretty much for a lot of people in the contaminated land worlds. And so let's talk a little bit about the background of PFAS. PFAS are a family of chemicals that were incorporated into a wide variety of consumer products since the 1950s. However, it wasn't really until 2001, so it's 50 years later, that we discovered that PFOS, PFXS, PFOA were present in nearly every living organism on the planet, and they're distributed widely across the planet, including areas with no known point sources. This led to an industry sort of phase out of these chemicals in 2002. So yeah, industry-led phase out of this chemical, which I'm going to provide some examples later where potentially they were not doing, acting in I guess, good faith is what you'd say. And so what we've seen is phase out of 2 chemicals and replacement with thousands of different chemicals, which is a really disheartening turn of events, particularly for people who, like me, who are engaged in this research area. Now, I don't want to come across that I'm anti-chemical, because I'm not, and it's important that we use chemistry in modern life. Modern life is, we expect it, and you know, we're all sitting here, probably we're on our computers or our iPads, our mobile phones, and all of the chemistry plays a central role in helping us use all of that technology, but we need to use it well, and we need to use it properly. And so there is some debate about the role of fluorotechnology in our society. And on the one hand, you have the industry saying that fluorotechnology is an essential for many aspects of modern life, and that is a totally true statement. And on the other hand, you have an international consensus statement saying that we call on the international community to cooperate in the limiting the production and use of PFAS and in developing safer non-fluorinated alternatives. So the things that you may know for your PFAS are things like your non-stick carpet, non-stick paint. You have Gore-Tex for your clothing. You have non-stick cookware, and your outdoor apparel is also full of fluorinated chemicals. And it's one of the few contaminants that there's a positive association with wealth. So the wealthier you are, the more likely you are to have these chemicals in your body. And so the story starts with C8 chemistries, PFAS, PFOA. They were the main chemistry that we used for a long time until it was discovered that they were accumulating in the human body. Their chemistry is different to persistent organic pollutants in that, or the first generation, in that they actually are water soluble and they move through the environment through different means. So the first generation, such as DDT, PCBs, and even the flame retardants, essentially they didn't like being in the water and they were accumulating in the fat. And so they were semi-volatile and move through the atmosphere and also bioaccumulate and biomagnify through food chains. PFAS moves in a different manner, and precursors, which I'll talk about in a second, have a role to play in how they move through the environment. And so this functional group, so they actually are both oil and water repellent, but the functional group gives them a degree of solubility. And so some of the impacts that we're seeing throughout Australia and throughout the entire world about particularly their contamination of drinking water it's because of the thinking that was applied from first generation, where they didn't think it would move through the soil profile into the groundwater, in my opinion. Okay, so these chemicals are persistent. As far as we know, there's no natural degradation process for them. They're mobile, they're bioaccumulative, and there's some debate about their carcinogenic potential. In recognition of all of these facts, PFOS was listed as a United Nations Stockholm Convention persistent organic pollutant in 2009, PFOA in 2019, and PFAS is currently under consideration. So the industry basically banned these two voluntarily and replaced them with families of C2 to C12 PFAAs, and these are the terms I'm going to start using, and PFCAs. So now we've gone from 2 to 28. But it gets worse, because what they then end up doing after that is we have per and poly. Per means it's fully fluorinated, and this is the terminal product that, like a PFOA or a PFOS. And then they started adding these R groups onto the PFAS, and you don't really need to be a chemist to sort of see that this side of the PFAS could probably be mineralized. And then that's exactly what happens. This side gets mineralized, and then we get left with the compounds that we think are toxic. So now we have an expanding universe of PFAS. We have over 4,000 different types of PFAS whereas in the past we only had 2. And so this poses a massive challenge for analytical chemistry. How do we monitor for 4,000 chemicals. How do we determine which are the priority chemicals, and how do we move forward in this in a meaningful way? And you know, some of the sources of PFAS to the environment include factories. So we don't typically manufacture PFAS, and as far as I'm aware, never have in Australia. But the Defence Force and airports become significant sources of PFAS, and they've become significant sources of PFAS primarily due to their use of AFFF. And we're going to need some new terminology in this space as well in time, because we're going to have diffuse background similar to lead occurring for PFAS for the foreseeable future. So your home environment is full of PFAS. Wastewater treatment discharges will always, and currently there will not, I'd be very surprised if you ever find a wastewater treatment plant that's not have PFAS in it at low levels. And landfill leachate, of course, is a repository for all the chemicals that we've used in society will end up there. Okay, so AFFF is one of the main sources of PFAS, and they're proprietary formulations that are used for extinguishing hydrocarbon and solvent fuels. Okay, so in terms of the regulations, we're on to NEMP number 2, and when the first one came out, and if you'll see this number here, which is less than 1 nanogram per litre, I don't think anyone in the country actually had the capabilities of measuring that low. So it's actually a significant challenge to be able to measure less than 1 nanogram per litre. Anyway, so this was just recently updated, and we still have the same very stringent guidelines. So I'll be comparing our results to this later, 1 nanogram per litre and then 130 nanograms per litre. Okay, the Footscray fire. The Footscray fire occurred on August 30th, 2018. It is an illicit stockpile of unknown chemical substances. It's thought that the barrels included ketones, phenols, PAHs, BTEX, and possibly PFAS. The fire, the length of the fire ranged from 3 to 8 days, and at its peak there was 16,000 litres per minute of fluorine-free foam was employed. The MFB, Melbourne Fire Brigade, states that they phased out AFFF in 2009, and all of this discharge water from the attempts to extinguish this fire ran off into Stony Creek. So there was a huge plume of toxic smoke that was released into the atmosphere, and the water that ran off into Stony Creek resulted in hundreds of dead aquatic life found dead, and it was primarily likely due to the solvents, not from PFAS. And EPA has reported that PFAS is one of several chemicals that were found in the creek following the fire incident. Okay, so I was actually in Poland when this happened, and Jay, who's going to talk later, in case anyone gets arrested, Jay, we'll say it's your fault. Okay, so Jay went out and got us samples, and we were going to do some targeted analysis on this, looking for PFAS, because that's what we have capabilities of doing in our lab. Okay, so Jay collected 15 samples. He collected 250 ml samples collected from 5 sites in triplicate. Okay, so this is a map of the Footscray area, and site 3 is the point source, and he collected 2 from upstream, and then we also collected 2 from downstream. So the samples were actually collected on the 1st of September in 2018, and that was, we took follow-up samples 2 months later on the 7th of November, Jay. Jay took samples 2 months later on the 7th of November. Okay, and one of the things you can see is that there's a clear discoloration coming in from site 3 through to site 5, which would indicate a bit of the ash and the chemicals that are being released into the environment. Okay, so we actually have the capabilities of measuring over 50. We have a targeted method using a triple quad I'll just talk about in a second. And I've made these ones on this side a little bit larger because these are the chemicals that we're going to sort of focus our attention on. So PFCAs are like PFOA, PFSAs, that's the PFOS. We have this other group, this is called an FTSA. These are like a 6-2 FTS. This was brought in to replace PFOS. And then when this one got banned, they brought in another compound that degrades into this, that degrades into this. Just hopefully that makes all sense. Anyway, so there we've got FTSAs. And then we also have DIPAPs as well, which are primarily used in for paper coating. Okay, so we use a technique called isotope dilution for our analytical approach. Isotope dilution is where you add a carbon labeled surrogate of all of the chemicals that you're interested in or near to into the sample prior to extraction. You use the carbon labeled isotopes to determine the amount of loss or that you've had or even enhancement during the LC mass spec phase. You need to, with this type of analysis, it's really easy to get contamination because PFAS is literally in everything. So you need to be really strict with your quality controls. And we typically do matrix spikes laboratory control samples, which is where we measure a known sample of known concentration as a sample and report its recovery, and a blank. And we have the capability of measuring below 1 nanogram per liter. Okay, so the way that the approach works is we have a 250 ml polypropylene bottle. We filter it through a glass fiber filter first. We add in our surrogates then, and then we're passing it through weak anion exchange SPE resin. And then what we do actually, you need to, for those people who do PFAS work, you need to actually wash your bottle with a little bit of methanol to get all of the PFAS that's stuck on the side of your bottle before pouring it through your SPE and eluting your PFAS off the SPE cartridge. I've got the solids extraction, but that wasn't actually included in this study. Okay, so here's some of the results. Okay, so what I'm actually going to show you now is there's two things on this one. This is the PFAS concentrations on time 1. And I've got the concentrations reported in terms of the groups of chemicals. So these were those 4 groups that I showed you earlier on. That's the color coding that we've got. So on the left-hand side, we have concentration, and on the right-hand side, we've got the percentage contribution. So what you can see is that, so I don't know if everyone's aware of this, but all of the surface waters in Melbourne have PFAS in them. So that there is just this background of PFAS coming through. Okay, then we have, this is the fire site. And what we have is PFCAs, PFSAs, and the FTSAs. And we start seeing this one as being, this group being the dominant PFAS that's present in this sample. And then we see this reduction occurring between sites 4 and sites 5 with time. And so this is just at time point 1. So how about we have a look at the 2 time points now? Okay, so this is actually, the top line here is concentration, and please note that we have, so this one here, and we have 2 different axes here. We have to 5,000 here and 1,000 here at time point 2. And you see that the sum total concentration reduces between these 2 times from about 4,100 to a bit over 800 nanograms per liter. What we also see is that the percentage contribution begins to change. Okay, so we start seeing that we get less FTSAs present in these in the sample at time point 2. But we still got elevated levels of PFAS 2 months later entering the water, and it still has an impact upon site 4 as well. Okay, so what are the chemicals of interest then? So the blue ones here are still all of your PFAAs. And probably the take-home message is this one here. So the main contributor was PFOA. So PFOA is the banned C8 chemistry, but it was found in reasonably high concentrations. The next one is PFOS. That top one there is PFOS. Again, the banned chemical is the main constituent of that we're finding in the water. And then PFAS. So the 3 that are Stockholm Convention are the three most dominant of these family of chemicals. And so we see that domination of the profile by those 3 chemicals, and it sort of reduces in time with in time 2. So they're significantly reduced. Okay, so for comparison, if PFAS was discharged through AFFF, there's actually almost no other studies like this in the scientific literature. But in 2002, there was reports of measuring the nearby creek in Canada, and that the levels that they report were closer 2,200,000 nanograms per litre. Now, it's really important to remember that pretty much anything prior to 2006 in the PFAS space is unreliable. And the reason it's unreliable is because they weren't really using carbon-labelled surrogates. They weren't present at that point. And you have a high level of background contamination, which they probably weren't aware of. So it's possible that it was actually that level, but it's possible that it wasn't. And the event that was most similar to what we encountered was a petrochemical fire in Japan. And actually they report reasonably similar levels to what we found in our study. Okay, and so the other one here that's important is this 6,2-FTSA. 6,2-FTSA is a replacement chemical for PFOS, and it's also a degradation product from other AFFF components. So so it's important to note that this one's significantly higher concentrations. Okay, and then the last piece of this targeted work I want to show is this chemical here, which is a chlorinated PFAS, and it's a replacement of PFOS, never been used in Australia, but somehow finding its way into a surface water sample down the river. Okay, so this is, in terms of our targeted work, I'm going to move on to, this is only the first half, and I'm going to rush through the second half. We found PFAS in 100% of the samples. PFOS, PFXS, and 6,2-FTSA accounted for the majority of the PFAS. 40% of the samples had PFOS exceeding the 95 species protection limit of 130 nanograms per litre. Okay, so we also would conclude that PFAS was released from the fire. Whether it was from the fire brigade or was stored in the chemical warehouse, we don't know, but we do believe that we have enough evidence to support the finding that PFAS was certainly released during the fire, and most likely released somehow from AFFF. Okay, so moving on to the bigger picture questions, which is what actually have these people been exposed to? So you may have seen this article in the newspaper where the firefighters, the men and women who have been basically risked their life to put this fire out, some have been reporting long-term health impacts. And so this is really where we need, as analytical chemists, think about how we move in a direction that provides more meaningful answers to these sorts of questions of what actually are we being exposed to. Because when we do traditional analytical chemistry, this type I've just shown you, is it's hypothesis driven. I'm deciding that I am going to look in this sample for these chemicals, rather than I'm going to determine what is in that sample. And so it's a paradigm shift that we need to think about. And
Brad,
yes,
just hold for a minute, because I need Richard Thwaites to answer the question that Jean Meeklim and others have put. It looks as though we're not able to get more people in than 100,
right.
which is a bit strange. Comments, Richard?
Yeah, I've been on the phone to Elise. The RACI paid to increase the license to 500, but that apparently hasn't been processed by Zoom yet. We are recording the seminar, and everyone who's registered, and if we can get the names of people who registered on the Eventbrite website, if Drew can send us those, we'll send a copy of the video to them. That's the best we can do, and it's very disappointing. We did anticipate a significantly more than 100 people, but the RACI was perhaps a bit slow in getting their increasing the size of their license. So sorry about that, but there's nothing I can really do about it at this juncture.
Thank you, Richard. And I'll mute myself and over to you again, Brad.
Sorry, Richard and Ian, if you would like to transfer the host to either Brad or myself, that'll go over to the UniMelb account. The link should stay the same, but we might be able to allow more people in using the University of Melbourne account.
Could you try that, Richard, please?
You just need to make me or Brad the host.
How do I do that?
So if you hover over my picture, and in the corner there'll be the 3 dots.
Hang on, which corner?
So can you see my video?
Yep.
3 dots, and then you make me host if you click on those 3 dots.
3 dots, there you are. How do I make you host?
There should be an option in the drop-down menu.
Where's the drop-down menu? He said naively.
Oh, if you just click on my face.
Click on your face. Hang on. Not working, just a minute.
How about I keep going? And Drew, why don't you send Ian your phone number?
Sure.
So Richard, your phone number.
Richard, I need to talk to Richard on that. Yeah.
Okay.
Thanks.
Okay.
Thanks, Brad.
No worries.
Sorry about that.
So we're up to the fun part, I guess, or hopefully the revolution in analytical chemistry. And now both of these things are necessary. You must have good quantitative data that currently really can only be generated through a triple quad. But the time of flight or high resolution mass spectrometry provides an opportunity to do new types of analysis that we haven't been able to do previously. Now, the main difference is that a triple quad is that I've decided what I want to measure for. For whatever bias that I have, I've decided that this is what I need to do. It's sensitive measurement, and I also need to build the technique for being able to measure the compound of interest. It's accurate, precise, reproducible, and it produces small data files. Now, the high-res approach actually contains a hell of a lot of information, and it sort of contains all of the information of all of the ions that were present in the sample that were ionizable. And so you also need to run it in multiple modes as well. You need to run it in ESI negative, ESI positive to get the different types of chemicals. And for the full complement, we will also need to start running GC QTOF as well to get the semi-volatile chemicals.
That's okay. That's 431.
So it's a high resolution technique, and we can use it for identification. It's a robust approach for unknowns. Now, the biggest issue is that it produces data files that are 3 gigs. And you need a powerful computational processing power in order to manage and process this data. Okay. So with that said, here's some QTOF data. Okay. So what we've done is we've done we've analyzed these samples, these extracts, in exactly the same way that we used the extract from the targeted work and we ran it on our QTOF. And this is really just a PCA of the features in that. And what you'll notice is, firstly, that in this PCA, we actually, these replicates are really tight, and we see that the Site 3 and Site 4 are actually really similar during time 1. This group over here is actually Site 1, Site 2 upstream, and both, they're very similar irrespective of the times of the analysis. And then we see that this is actually Site 3, Site 4, Site 5 at time 2. So we start seeing these sort of, still this separation, and this last group here is actually Site 5. Okay, so what we get is this raw sort of intensity mass spectrum, and we can get up to 50,000 different, they're called sort of features or entities, and we're trying to process this in a really robust way. Okay, so when we process the data, we have made some really stringent rules on the quality of the data. So we've said that the entity must be present in all 3 of the replicates. And we try to get also, it's called MS spectra on this, so we can actually do some confirmation on the identity of the chemicals. So what we got here on the right-hand side, the left-hand side is what we have the number of entities. And so you'll see that the blue side is time 1 and the green is time 2. And you'll see that there's more entities in the water in those samples at time 1. So we have a total of 8,200 and 6,250 at time 2. And on the right-hand side, we have a heat map, okay? And on the heat map, you can see that the red indicates all of these lines are a different chemical, essentially. They're all different chemicals. And on the right-hand side, you'll see that time 1 and Site 3, time 1, Site 4, and time 1, Site 5 have more red lines indicating there's more entities at higher abundance in these samples. Okay, so the first, what we need to do is figure out how, we've got such a huge amount of data, how do we figure out what's the most significant? And so I've done some unique features as well. So I want to look at, this is a Venn diagram looking at the number of unique entities comparing time 1 and time 2. And what you'll notice here is actually there's nothing unique in time 2. So what this means is, in terms of identifying, which is really all we're going to do with the QTOF work, is that I can just focus my attention really onto the time 1 data samples. And this will also give us, in many cases, the more intense peaks, which are assuming correlates to the most concentrated in the sample. So it's this number here that we're thinking will correlate to these peaks that are the significant ones that we need to follow up. However, it's worth pointing out that it's not necessarily the case that the unique features means that it's the chemical of most importance, because PFAS, as an example, was present in all of the water samples, Site 1, Site 2. So you've got to be careful with this as well. Okay, so what we see, this is a raw spectrum on the right-hand side, and we start seeing thousands of chemicals. So this Site 1, Site 2, clearly not present. These same chemicals, we see, this is just raw spectra. I don't know what it is. That's the high resolution mass spec number. But we can see there's literally thousands of these chemicals that increase in Site 3 and are present from Site 3 onwards. And so the raw numbers, we get in 1,000, and we get more entities at Site 4 than Site 5. And roughly speaking, we sort of have 2 groups, and Site 1, 2, and then Site 3, 4, and 5. If we run PCA again on this, just on this time point, and what we see is actually we've got same sort of grouping. Site 1, Site 2, no difference. Site 3 and 4, there's no difference during the incident. And then we have Site 5 that's a little bit different. Okay, so if we combine a Venn diagram with the PCA to try to find out where to, the key thing is, where do we focus our attention with so much data? Now, okay, so both, and what I've done is I've color coded this. So that's the PCAs on the right hand and then the Venn diagrams here. So the Venn diagram won't handle 5 sites. It can only handle 3 or 4. So what I've done is I've done a Venn diagram of Site 1, 3, and 5, and 2, 4, and 2, 3, and 4. What you can see is there's very few unique features in site 1 and 2. And so therefore it's probably not worth really investigating them too much. But what we can actually see is there's huge overlap in these chemicals in site 3 and 4, and big numbers too. And that's probably where we sort of need to focus our attention. And then site 5 has a number of unique entities, but it's probably, in terms of figuring out what came from this fire, this region, so we're narrowing it down from 8,000 to 4,000. Okay, one of the ways we can do this is a technique called suspect screening, where we scan the raw QTOF data against, they're called PCDLs, Personal Compound Database and Library Managers. We have 3 that we've scanned our data against. US EPA PFAS priority list has 365 chemicals, pesticides, and another water contaminant database. The idea is actually that you match, you get increasing levels of confirmation as you match high-resolution mass data with MS spectra and retention time if possible. Okay, so screening this against PFAS. So it's not a great chromatogram, but all of these are the unique PFASs. So I just wanted to show that it's possible. So when you actually just do a raw screen of one sample, this is just one of the samples, against the database, we're finding all of the hits from our targeted work are present. So proving that this technique is actually capable of determining the presence of these different chemicals. But you'll also notice there's all these other things in here that we may or may not have analytical techniques for. And so here are just some of the examples of the different types of PFAS. Some of these I've never ever seen before. Now these are potential. We still have to confirm them. So these are some of the suspects that have been identified. And this is sort of the profile that we were getting from a lot of these different chemicals. Okay, so the way that you have to then build the method is, if you find it in a QTOF workflow, you need to actually then go and validate it with purchasing a standard and the confirming retention time and mass spectrum at the current process. So there's this sort of cycle loop around this. And this is just an example where we've done it in the past. It's not in this study actually, but for 6.2-FTS, and we purchased a standard, confirm mass spectra, confirm retention time, and therefore we can confirm that this is definitely present in that sample. I want to just provide a little bit of more information about how complex this type of analysis is. So you see this little slither? I've just zoomed in on that little slither. That's all of the chemicals that are unique chemicals. And this is after we've applied our filters. And so you'll just see there's a lot of numbers there. And that means that we don't even have a tentative identification for what this chemical is based upon a screening against our database. And the last thing, so when we screen against our database, here's some of the chemical names. They won't provide any structures. Here's some of the chemical names that we potentially are in this water, and it includes pesticides, drugs, and I'm not going to go any further into that because we still need to confirm that that's actually what's there. So we have higher confidence in about 4% of the identities, only about 320, and low confidence in 23 of the identities. And so in terms of this type of work and the approach, quoting Donald Rumsfeld, we had known knowns, which is where you use your triple quad, and then you had known unknowns, and that's where you use a suspect screening database. And then you have unknown unknowns, and that's where we're really just interrogating that QTOF file and we're going to do formula generation on that in time. And it's this big loop that you need to use, this is where we have to do quantitation. It has to be the triple quad, and then you find the new priority important chemicals through the QTOF. We have a significant challenge ahead of us. This is the number of unique chemicals registered in the CAS database, 147 million. And we are not learning historical lessons, and we are still introducing new chemicals into the market routinely that were likely to be persistent, bioaccumulative, and toxic. So with that, I just want to thank the team, Drew, Suba, Jay, Rob, Tori, Shiwei, Julia, and Maddie. And thank you very much for listening to me talk tonight. And I think I'm taking questions, but I'll wait I'm not sure if that's what Ian's...
Indeed. Thank you
Yes?
so much, Brad, and everyone clap or indicate thank you in that other way, which I can't remember. So well done, Brad. That was fantastic. I think we're still locked in at 100, though, aren't we, Brad, Richard and Drew?
Yeah, we've given Drew the job of being the host, but I don't think he's been able to increase the participation rate, which is a pity. But as I said before, we're recording it, and we will, if Drew can send us or send Elise or me the list of everyone who's registered by Eventbrite, we will give them copies of the video of the seminar. Best I can do.
Is that okay, Drew? Sounds good?
Yep, sounds good to me.
And apologise that although the RACI told me, and I'm sure they did, increase their licence to allow 500, doesn't seem to have been accepted yet by the Zoom organisation.
Okay, Richard, thank you. And I let you go on, Brad, purposely because I wanted you to present everything that you had. Very complicated and well done, thank you. Questions for Brad Clark, please, anyone? And I'm chairing but not hosting, so Drew, be ready to watch hands going up.
I'll ask a question. Hey Brad, thanks. Great presentation. I wanted to ask, with a lot of these new things that you're finding in the untargeted screening, do you think they're compounds that have been released as is, or would they be breakdown products of other things? And is there any way that you can kind of trace them back to understand what their origin was, and if they're breakdown or whether they're as they were? Do you know what I mean?
Sort of. I think it's a really good question. I think the biggest challenge that we face, the first challenge is getting identifications, and it's not as easy as it sort of looks in terms of just confirming. So really that technique is using presence or absence, and we need to make that move forward into doing semi-quantitative work. In the PFAS example, it's pretty clear that some of those things that we found are actually the reason that the 6,2 FDS is, or it's very likely that they're the primary source of the 6,2 FDS, which then becomes the primary source of the carboxylic acids. In terms of doing more backtracking, because obviously also there's all these combustion products in the water too. That's going to be a significant challenge. So we're going to continue doing some work in that on those extracts.
Yeah, that's great. Yeah, thank you. Cheers.
And another question, please. Anyone?
Ian, you know the rules. You're meant to ask the question.
Yeah. Jeff Simpson, how have the emergency services responded to the fact that you've found all this PFAS in the foam, in the actual samples, which would have to have come out of the foams that were used? Has that actually been followed through at all?
The EPA acknowledges that the PFAS is been released into the water. I don't know if they know the exact source. I mean, it could also be that the PFAS is stuck on the firefighting trucks. You know what I mean? Like, and then they got potentially they got fluorine-free foams that they've put into the firefighting trucks, and that's still just working straight through the system. You know, to me, this seems like a legitimate place to actually use PFAS. You know, we want to put out a toxic fire pretty quickly, and it's one of the best things you can do to put it out. Rather than just doing a training exercise to lighting jet fuel and then setting it and then putting it out. So, yeah. So where it came from, I don't know. What the EPA's response is going to be, I don't know either.
Thanks for that.
I think Bob's going to answer the question.
Okay, I have a comment, but I won't make it if there are. I'll make it if there are no other questions. Let's have another question.
Yeah, can I?
So
Roy, Bob Simons here.
Roy Tasker, could I ask a question?
Roy Tasker, go ahead, please.
Yes. G'day. I'm from Planet Ark, and we're doing a report on PFAS for the general public. And of course, what they're most interested in is the toxicology of this. I mean, it's frightening to think of chemicals being accumulated in the body and all that sort of thing. And so I'm trying to work out what to say about risk. I mean, it isn't as if people can stop their exposure to these things.
Yeah, I think it's a big.
Yeah, sorry. Yeah. Can you give me any idea on the latest on the toxicology of all this?
Well, actually, I won't talk about the toxicology because Jay's going to do that. But what I will make a comment about is it's actually bigger than individuals. And the reason I was sort of showing the examples is because we're not learning our lessons. And what we're doing is making bigger problems when we regulate one. And we also need to be nuanced in this discussion as well. So for example, a lot of people are upset about the West Cape Bridge tunnel spill going out. You're exposed to PFAS in your home. You're exposed to PFAS probably when you have pizza, when you get a takeaway coffee cup. Your clothing all has PFAS in it. So we need to have a more reasonable discussion about these exposures, and then as a society decide about what chemicals are important for us to use. And so I don't think it, personally, I don't see that we should be divesting that down to the individual. And I think we should be not introducing 4,000 chemicals that have with unknown effects in the environment and on human health. And I think the main thing that we can do moving forward is change the burden of proof. And the burden of proof should be put onto the chemical manufacturers that this chemical will biodegrade, it doesn't bioaccumulate in the human body, and it's non-toxic. And if they can't satisfy that, similar to the way pharmaceuticals get registered, they shouldn't be allowed to be produced.
I'm right with you. Thank you.
Thank you, Roy. Thank you, Brad. Another question?
Brad,
Yeah,
I've got a question.
We're going to be over time, but don't worry, please. Who was that? Who?
Brad, Luke McGuinness from Western Australia. We've banned PFAS from moving across the border to WA, of course. Sorry, so has that creek been routinely analysed in the past before the fire?
Yeah, I mean, there was I think the levels that were, no, it wasn't routinely analysed, but we have seen some PFAS levels, and they're pretty similar to the Site 1, Site 2. Yeah.
Can I just add a comment? So Bob Simons from Eurofins, we analysed some of the samples from that fire as well. I want to confirm with what Brad said, that there could be some, that the fluorine-free foams were used by the MFB to fight the fires, and there could be some residual materials there, but some communication with the Vic EPA suggested that there were some drums of PFAS or AFFF stored at that fire, and that's where the major release had come from.
Thank you for that, Bob. Much appreciated. One more question, I think, if anyone has one. If not, I will just mention that I find it fascinating that the human race is now considering modifying the atmosphere of Mars using sulfur hexafluoride or octafluoropropane. My God, if we can't manage this planet, let's not spoil another one just yet, eh? Thank you, everyone. And now let's have Jay, please, Jay Makiandi, who will be speaking on characterising the effects of PFASs on health and disease, an opportunity for exposomics or exposomics, as he says. Jay.
Yep, super easy. Sorry, I wasn't prepared to speak second. Hi everyone, my name's Jay. I'm a first year PhD student, and my project is looking at the relationship between environmental pollutants and human infertility. But today, I'm going to be speaking to you about a relatively new concept called the exosome, and I'm going to be discussing its implementation into human health research into PFASs. So I'm sure there's some of you that don't understand or don't know what the exosome is. It's quite a new term. I'm just going to give a brief background review of it. So in 2005, Dr. Christopher Wild, the former director of the IARC, championed the need for a comprehensive exposure analog to complement the genome. He wanted to redefine epidemiologic findings and public health research. Initially, science and medicine were slow to respond to the change because by their definition, it seemed was confined to exposure assessment. As such, in the following years, the definition, Christopher Wilde defined it as that which encompasses life course environmental exposure, including lifestyle factors from the prenatal period onwards. In 2014, Miller and Jones expanded the definition to include the cumulative measure of environmental influences and associated biological responses throughout the lifespan, including exposures from the environment, diet, behavior, and endogenous processes. So there's two broad domains in the exosome where exposures can arise from. There's the external part, which is just things you're exposed to in daily life from diet, soil, behavior, lifestyle, etc. Then there's also internal exposures as well. Generally through the internal exposures are generally markers of external exposure. The difficult part about exosomics is how do we characterize external exposures and the internal responses? So the interpretation requires an array of analytical tools. They're generally complemented by each other. So omics technologies does require bioinformatics and biostatistics. And when you're looking at tens of thousands of participants, you will need artificial intelligence to prioritize the findings to pinpoint the ones that are most relevant. So there's two general approaches to the exosome. First is a targeted or a bottom-up approach. This is essentially what research has done in the past. It's just choosing a set of exposures or environmental compartments, such as air, food, water, and determining the biological pathways by which such exposures will lead to disease. The other approach is called the top-down approach. This is more of an agnostic view where you don't rely on external information or previous data. When people think of exosomics, they generally do think that most of it's agnostic, that you're doing some QTOF work, for example, and just seeing what's there, and then you prioritize the findings. So it's important to note that the exosome isn't a replacement of traditional reductionist research, rather it's complementary. So the exosome sort of builds on previous research while retaining some of the previous findings as well. So exosomics generally employs mass spectrometry, from omics approaches, but also wearables as well. So wristbands to determine, airborne chemicals, geographic sensors, etc. And yeah, so let's go to PFAS. So you've heard a bit of PFAS from Brad. Basically, with the PFAS have been a global problem since for the past few decades. It's only been a recent problem in Australia for the past probably 10 years or so. But now there's a lot more articles, especially after the Footscray event, to suggest that PFAS are very bad in Australia. I'm not sure if any of you are aware, but I think Erin Brockovich came to Australia last year, and she's gathering people from these affected communities to launch the largest class action lawsuit against the government for PFAS contamination. And these people are generally from these areas here. So in areas where there's a Department of Defense site or a firefighting training site, historically they've employed HLF or firefighting firms for their exercises. What's happened is the PFASs from these have percolated into the groundwater, which then the residents will consume either by drinking water or via food from contaminated food that the cows have drunk. And regulatory agencies have come out and told the residents, not to eat the eggs or not to drink the water and not to eat the cows, for example. So it is a very pervasive issue in Australia. In terms of toxicology, PFASs are classified as a multi-system toxicant. If any of you are aware of the toxicity of lead, essentially there's multiple targets of toxicity. These aren't all the health effects that are associated with PFAS. I thought I would try and narrow it down. But PFASs have also been associated with neurological problems, but with issues in epidemiology and questionnaires, the toxicity or toxicology is mixed. In terms of the most comprehensive data we have on PFASs, it would have to be from the See It Health project. So I'm not sure if any of you have seen the Dark Waters movie. It recently came out. I haven't seen it yet. I really want to. But that movie talks about the DuPont manufacturing plant and how from the years, I think from 1950s to early 2000s, DuPont was just discharging PFOA into the air and into the ground. And what was happening was it would percolate into the ground, go into the groundwater, And in these areas here that you can see in red, well, actually most of these areas, they were seeing a high incidence of various diseases. So what happened was, I still haven't seen the movie, but so they were found out for this, and then they relaunched the See It Health project. So they looked at 69,000 participants between 2005 and 2006, and these are the results here. So serum PFOA levels were around 33 nanograms per mil. A recent report that came out in 2018 from the CDC suggests that geometric means for a normal population in America is around 5 nanograms per mil. So it's still very high. And the most important findings were they found, this is probably the most concrete evidence for a health effect with PFAS exposure and a health outcome. They found that PFOA was associated with high cholesterol, thyroid disease, ulcerative colitis, pregnancy-induced hypertension, and kidney and testicular cancer. So in terms of health effects, I would say these are probably the main ones. Despite the substantial progress made to better understand the hazards that may be inherent in PFASs, there are several challenges to identifying their associated health risks. First is that we don't know the individual health effects for the majority of PFASs. We've only looked at a subset of PFASs or perfluoroalkyl acids, such as PFAS, PFOA, or perfluorohexane sulfonate. These are only a small group of PFASs out of nearly 5,000 now. So if we are to fully characterize the health effects of PFASs, we need to broaden our range, and we can do that by some untargeted work. Another issue is that polyfluoroalkyl precursors are overlooked despite their ability to degrade in the body. Like Brad mentioned, a key compound found in the first-grade waters was 6,2-FTSA. This is an intermediate, or it can be a precursor as well, but this compound degrades in the environment down to some perfluoroalkyl acids. If we're ignoring these, then we're ignoring what they can degrade into and the effects that are associated with that compound. The second is that epidemiology and toxicology tend to focus on a single or limited number of compounds rather than a mixture-based approach that more realistically resembles human exposure. A few studies have looked at target analysis of PFASs and are finding that we can only find up to around 85% of the extractable organic fluorine, suggesting that there's still much more PFASs in human serum that we're not aware of. Another issue is that we ignore the potential combinatory effects. We have additive effects, synergism or antagonism between PFASs and other chemical groups of exposure. The third is that while the biological effects while the toxicity is biologically plausible in that PFASs are responsive to a receptor in both humans and animals, the underlying molecular mechanisms are still unclear. We found that, well, with PFASs, there are significant differences between rodent models and human models when we're looking at, when we're doing mechanistic studies. And I'm just going to give a few examples here. So I'm not sure if any of you are biologists, but basically on the left side here with rodents, these are the traditional receptors that investigators looked at. They're generally metabolic, involved in metabolism. And what we were seeing was that while cholesterol levels in humans who were going up, for example, in rodent models are going down. So it was hard for regulators to establish consensus on biologically plausible events that suggest that PFASs are indeed toxic. However, we have found that there are new receptors on the right here that are more related to human health. However, these receptors have only been tested with PFAS and PFOA alone. So we need to test many more PFASs to make sure that it's not just related to them. And just on that point before, it's all about the translation between rodent models to human models. And as we'll see in rodents, they were seeing a decrease in serum cholesterol and triglycerides, whereas in humans, we're seeing an increase. So this is what was stymieing the consensus. Epidemiology is also without its flaws. We see that with cross-sectional designs, for example, there have been hundreds, nearly thousands of studies of PFASs that look with cross-sectional designs. However, looking at one point and then coming back to it a couple of years later, everything that's happened at that time it precludes any sort of causality between that. So it's okay to say there's an association. However, there are so many events that could have happened in that time that could make it so that PFASs weren't the cause of the observed health effect, for example. The longitudinal design is the gold standard for epidemiology. However, if we apply a priori consideration to it, that we only pick a few compounds, we're ignoring other potential co-exposures that could be mediating the same effect. And then we also bias confounding and statistical power that are related to it. With the bias, obviously, like I said before, if we're choosing certain exposures to look at, that's a bias. We're saying, okay, we're just gonna pick at these ones and ignore the rest. And then statistical power, it is hard to recruit a lot of people, especially in the hundreds or thousands, because it's very costly. There have been a few studies that have done this, but even still, it's very varied with population size. So is exosomics the solution? So exosomics, to apply an exosome approach, it requires multidisciplinary collaboration by unifying the strengths of different scientific domains. By this, and also data sharing as well, and data availability, we can compile all the data into a central database. And if this database is open access, then researchers all around the world and other research teams can access this, and they can investigate PFASs in their field of area. And there's a multitude of outcomes as well that come about this, ranging from public health or even mechanistic insights and management strategies. In terms of what we know from exosomic studies and PFASs, there isn't a whole lot yet, just because it's quite an effort to look at hundreds of thousands of features and that narrow it down. The HELIX study, the Human Early Life Exosome Study, was finished in 2018, and it looked at 32,000 mother-child pairs from 6 European birth cohorts, which is supplemented in with new. Sorry, I think my AirPods are about to run out. Sorry about this. We'll see if it switches to my computer. But so far, we've only found that there's only been around 3 studies that looked at PFASs and the exosome. And we found things ranging from the PFAS and PFNA, so the C8 and C9 PFSA, are associated with lung impairment, impairment of lung function. And we've also found some potential interactions with endogenous exposures, as well as other exogenous or external exposures such as PCBs. And this one here, the human biomonitoring 1 value, is from the Human Biomonitoring Commission in Germany, which just says that the levels of these compounds were above the threshold, which would equate to adverse effects. There have been many developments in the field of exosomics. Like I said before, with the HELIX study finishing in 2018, many studies are coming out now and analyzing the data and finding new results every year.
Sorry, can everyone still hear me? Okay, cool. Sorry, my AirPods just ran out. So new data is coming out lots of new data is coming out every year, and they're doing these network studies and finding possible interactions between different chemical species and other endogenous metabolites. I would like to take this time to boast a little bit. I was working my presentation last night, and around 2 AM I got the email saying that it had finally been published. So all this work has been the combination of nearly a year's worth of research, 200 peer-reviewed studies, reading, and it's been a long journey, but I wouldn't say I'm an expert yet, but I know a fair bit about the topic. And thank you very much. I would really like to thank Dr. Bradley Clark and Dr. Mark Green for all their help, as well as Dr. Sonia Dagnino and Dr. Tarun Animal for their help with the analytical side of things in Exosome. Thank you.
Thank you very much, Jay, and let's have everyone clap or whatever to say thank you. I'd like to put a question to you first, if I may. I don't understand. When you say exposome quickly, it sounds...
trouble. Okay, can hear you now.
When you say exposome quickly, it sounds like exosome, and exosome is something completely different, isn't it? Something to do with RNA. So all of your talk means exposure with the P.
Exposome, yeah, like exposure.
Yeah, and I don't understand it all yet, but I've got it on the web. So, and I hope we'll get your talk too as attendees. We would like to have your talk so that we can look at it at our leisure as well.
Yeah, I apologize. It's hard to talk about a whole new concept and its implications.
Yeah, absolutely, absolutely. I'm not being critical, for heaven's sake. And if Brad's okay with Brad's presentation, your presentation, and the next one to be available to us all, that would be delightful. Is that all right, Brad? He's nodding. Good. Okay. Right. Questions, please, for Jay.
Could I add a very quick question? This is a sensational talk for what I need at the moment. I'd love to get a list of the references there. I know you've got the citations in your presentation. Could you possibly provide the actual references themselves? It would save me an enormous amount of time.
Yeah, that's okay. I could send you a link to the article that's in press at the moment. That article has all the references there as well. All those references were they were a meta-analysis and also longitudinal studies, so it's not just a cherry-picked cross-sectional study.
Fabulous. Thanks, Joe.
Good show, Roy. Thank you for that. Those references are listed, but does that mean we can click on them and open them, Joe?
If you would access the article, you can, yes.
Okay, because my difficulty now, having finished my research, is that I've lost that facility through the university to access learned works, and I'm trying to get it again because I've not stopped researching. But anyway, next question, please, for Jay. If all quiet, thank you again, Jay, very much. And let's have, oh God, I've covered up his name. I've covered up your name. Where are you? Silly me. I've done something with something. Oh, there you are. There you are.
Szabo, it's the guy that we're going to listen to now, Ian.
Drew Szabo. Yeah, I'd covered it up. I hadn't lost it. Okay, so Drew Szabo of the University of Melbourne is going to talk about targeted analysis of per and polyfluoroalkyl substances in fledgling shearwaters from a remote Pacific island in Australia. And I was going to say, I hope that's going to be Lord Howe Island again, which there's been some very good television coverage of recently in environmental terms. So thank you. Please go, Drew.
Thanks, Ian. Yeah, that's right. And another PFAS talk. So hopefully this doesn't get too boring for everybody, but I'll try and go through all the chemistry pretty quick and just get to the crux of it. Yeah, so thanks for introducing my talk. I'd just like to thank Jennifer Labers. I think she's on here tonight. So she's the lead researcher with the Adrift Lab, and she collected all these samples for me last year from Lord Howe Island. And also Raoul Mulder, Mark Green, and Brad Clark, who are my supervisors at the University of Melbourne. Okay, so this is beautiful Lord Howe Island. It's about 800 kilometres off the coast, east coast of Australia. It's a relatively small island, only about 14.5 square kilometres. And it was made famous in 2001 when they rediscovered the Lord Howe Island stick insect, which was thought to have been made extinct in 1918 after a boat was shipwrecked there by accident. Recently, BirdLife International has classed it as an endemic bird area with only 13 species of birds only found on this island, which is incredible. Now, 2 of these species are the wedge-tailed shearwaters and the flesh-footed shearwater. Now, both of these species are, just like all marine birds, in decline all around the world, or their populations are in decline all around the world, because they're impacted by things like bycatch, habitat loss, competition for resources, and solid waste pollution. Now, Jen Levers and her team have been studying this population for 20 years, almost 20 years, and they found things like, their populations have been impacted by climate change because they've found their shifts in foraging behaviour. They've also found elevated levels of heavy metals or metals like mercury, copper, zinc, arsenic, and cadmium, the lack of which have been increasing over that time. So why do we use marine seabirds as biomonitors? Well, they feed over large areas, and they also feed over multiple trophic levels. So that makes them an ideal candidate to monitor pollution in the environment, in the water, in the fish stocks, and even by proxy, things like climate change. And so Lord Howe Island in Australia is a relatively pristine, remote Pacific island. which makes it good for a baseline assessment for long-range anthropogenic impacts. So the diagram on the right there, you can see the 2 black dots are breeding colonies for shearwaters in Europe and Africa. And these birds will travel up to 400 kilometers away from their nesting sites to feed for their young. They don't always bring back food though, and this is the problem. So plastics can be found in 90% of fledgling birds' stomachs, which is just crazy. In one bird found last year, up to 116 individual pieces of plastic were found, weighing roughly 10% of the total body mass for that animal. Trace metals and persistent organic pollutants will absorb to marine plastics, leading to dietary exposure. And what we're interested in is, we know that PFAS also swarm to marine plastics, but we don't yet know what the contribution is for plastics leading to PFAS exposure in these birds. This slide might look pretty familiar, so I'm going to go through it pretty quick, but suffice to say that we know that the biological half-life of PFASs in birds is greater than 4 years, and in the environment, you know, it's estimated to be greater than 96 years. Yeah. Okay, so here's a small table of some of the laboratory toxicology studies that have been done. And so the first thing you'll notice is that PFAS is the most studied compound. And they tend to use, you know, the lethal dose endpoints. But the problem is the concentrations are not environmentally relevant. So the acute toxicity is much higher than the concentrations found in the environment. However, what I've highlighted in red here is the sublethal impacts. For example, the hatchability of chicken eggs may occur at environmentally relevant concentrations. As low as 100 nanograms per gram has shown a low adverse effect level in chickens, and that's what we're worried about. Okay, so the aim of my study was to analyze PFAS from the liver samples of juvenile shearwaters. And the reason we use livers is because, unlike persistent organic pollutants in the past, like Brad was saying earlier, that you know, in the fat tissues or adipose tissue, PFAS are mostly found in protein-rich tissues such as liver and blood. So they're the most ideal tissues to study. We also compared the concentrations of these PFAS with physiological fitness parameters, and also with the concentration of, or the mass of the plastic that was found in their stomachs, to see if plastic was a source of PFAS to these birds. So how do we do it? Well, we had 42 birds all up, which were all sampled within an hour of death. Each of the birds were all around the same age, between 80 and 90 days old. We took those livers in the lab and homogenized them. We used acetonitrile to extract the PFAS, and then a number of reagents just to clean up the extract before it was injected by LCMS, MS, and the concentrations of 50 compounds determined. So this, again, was all done by isotope dilution. So we introduced 14 mass-labeled internal standards, and we report the recoveries of these, and we have a certain percentage range around which we qualify as a true value. So of those 42 shearwaters that were collected, plastic was found in 70% of the birds. So there was 33 flesh-footed shearwaters and 9 wedge-tailed shearwaters. And even though they had the same body mass, the flesh-footed shearwaters were actually slightly larger in the head-to-bill length, the wing chord length, and the Coleman length. Okay, so here's a large and confusing table of the concentrations of PFAS, all 50 compounds. Now down the bottom, I've color-coded the different functional groups to pick out the easier ones. So here in blue, we have the carboxylic acids, including PFAR. And in red, we have the sulfonic acids, including PFOS. Now, to make it a little bit easier, I've just taken out those compounds that weren't detected in any birds. Oh, before I do that, I'd just like to make note that PFAR, which Brad and Jay were just saying is found in nearly all other wildlife and humans, is not present in birds. Okay, so here are the 20 compounds that were detected in birds. So we can see that PFAS is still there, and these 4 here are long chain carboxylic acids. So from 9 to 13, essentially, we have, that's the carbon length, 9 to 13. So because they were detected in more than 50% of our samples, I'm only going to be talking about these for the data analysis. Okay, so what did we find? Well, PFAS was significantly greater in concentration compared to these long chain carboxylic acids, but there wasn't any difference between the concentrations in the 2 different species that we looked at. Okay. All right, and now this is a comparison with literature found around the world. Now, this is actually a complete list of all the literature for PFAS detected in juvenile birds. So first thing you'll notice is it's very short list. In Australia, this is the first study of birds, juvenile birds in Australia, and in fact, the second paper or the second report of concentrations of PFAS in birds in Australia. So the rest are all being conducted in the Northern Hemisphere, and you'll notice that the concentrations are far greater in these studies that have been reported, and that's due to the fact that they were focused on areas of known contamination in the first place. So they were exposed to higher levels of PFAS in the first place. And we have the same kind of relationship between the concentration of PFOA and the carboxylic acids. So around the world, the carboxylic acids are found in lower concentrations as well. Another interesting point is that PFOA is commonly absent from birds around the world, which is in contrast to other mammals, including humans. Okay, so we found that the principal component analysis separated out the 2 species, and that was because of the physiological parameters mainly. But we didn't find a correlation between the concentrations of PFOS, PFNA, PFTA, or PFUDA with any fitness parameters. We did, however, find a weak correlation between PFTRDA, which is a 13 carbon length, and the body mass. And this is also, has also been found in kittiwakes from Svalbard. And they found that there was a correlation between PFTRDA and cortisol levels, which is a hormone that can have physiological impacts. All right. The good news is there's no correlation between PFAS concentrations and the massive plastic found in their stomach. So we're pretty confident that plastic is not contributing to the dietary exposure of juvenile shearwaters. Instead, we think it's just coming from the diet, the food that they're actually getting from their parents instead. Similarly, there was no relationship between fatty acid composition and plastic mass in these same individuals by another paper by Peter Pustik in Jennifer Laver's lab. And also these same samples have been sent to another institution, RMIT, and they're doing some work on some other persistent organic pollutants, which I'd be keen to see in the future. So where to now? Well, we still don't know the risk of PFAS to the adult birds in this population. So they're obviously being exposed because the juveniles are through their diet, but we just don't know their concentrations or therefore their risk. Overall, there's a severe lack of literature, peer-reviewed literature in the Southern Hemisphere surrounding PFAS and birds. I'll be looking at in the future concentrations of PFAS in birds near known contaminated sites, such as wastewater treatment plants and defence sites, which are more at risk, and the birds have received a higher exposure to birds, you know, compared to those in Lord Howe Island. And here's an example of a poster that the EPA put up near Hospital Swamp that recommends hunters not eat waterfowl, fish, from contaminated sites, because it may pose a risk to human health. Well, the concentrations in those areas were so high that there may be a risk to human health as well. I'd like to thank the research team, the Australian Laboratory for Emerging Contaminants, the Adrift Lab and the Detached Cultural Organisation, and L. Bryce for the funding for the field trip to Lord Howe Island, and Melbourne Water and Water Research Australia for helping my PhD project. Thanks.
I hadn't unmuted myself in time, but thank you so much. Thank you indeed. Please, everyone clap and say thank you. Well done. That was fantastic, Drew. Questions for you. I know Lord Howe Island because my wife and I went there in 1970 something by flying boat, and regard it as a shame that didn't remain as the only method of going there. Just to mention that the shearwaters are known over there and commonly as mutton birds, of course.
That's right. Yeah.
But let's have some questions for Drew. First class. Oh, that's surprising. I think it might be, I don't know how many, let's have a look. We've still got 73 participants. I like the idea of going into breakout groups. I don't know if, Drew, do you know how to do that, or Richard? It might be a good idea for us.
Drew's got the, so there we go. Drew's got the, sorry, am I muted? No, I'm not. Drew's got the host thing, so he may be able to get us into breakout groups.
Sure.
Not confident.
Can I just ask Drew a question before we jump into there? Sorry.
Yeah.
Sorry, it's Scott here from Macquarie. Drew, you only found the long chains. You know reason why there mightn't be the short chain PFASs?
Yeah, so the short chain PFASs just tend not to magnify in high order species like the long chains do. Yeah, so the chemistry of the longer chain means that it's like more likely to stay in the bodies of these animals. And because marine seabirds are at the apex predators, they're only receiving that high trophic transfer, yeah, from the environment. So some of those shorter chain compounds get filtered out, yeah.
Yeah, over time, yeah, sure. And did you look at the prey species that they were for PFAS as well?
No. There are a number of other projects that I will be looking at the diets as well and finding out what the relationship is between, their sources of food and, their level of exposure. But that's still in the works, yeah.
Okay, cool, thanks.
Nice.
Thank you so much. So very, very much. And let's, Drew, go into little chat groups then, if you would.
Sure, how many people? Per group.
How many people per chat? There's not much point if there are too many of us in. It just won't work. But does any? I think it's a good idea. I just never done it with so many. With
well, the numbers have gone down to 36 at the moment.
36, yeah.
Yeah.
I can do about 7 rooms or 6 rooms of about 4 or 5 people.
6 at 6, I suppose. Yeah.
Yeah. Okay. And we don't have to be too long. Then we go back to what sort of time shall we have per chat group? 5 minutes? No, 5 minutes or less or something with a warning. Yeah.
Sure.
Okay.
Okay. Here we go.
Nice job, Drew.
Yeah, awesome, Drew. Well done.
Yeah, really good job. I think I'm ready to hang up.
Here we are. Yeah.
Coming back.
Where's Ian gone?
Okay, I've got to go. Thank you.
Right, Roy. Good to see you.
See ya.
Thanks a lot.
See you later, Roy.
Bye.
See you, everybody, and that was great to do, indeed.
Where are you, Ian? Oh, there you are.
Yeah. Thank you so much, everybody. not time for other breakouts?
Not really, I don't think.
I think
No, only 17 of us left. So,
Yeah.
so we're getting the message. But all right, please, Brad, let's have all of the publications from today. You know what I mean? The talks for today, the publishers and so on, for each of us, if that is at all possible. Is that all right, Brad?
So what are you asking?
Well, like I did earlier.
What, the presentations?
Yeah.
We'll get the
Yeah, that's no.
Let's have them if we may.
That's no problem.
Good show.
Sure, we can
Good
do
show.
it, yeah.
Thank you. Thank you. We'll talk some more after. Thank you, everybody. And you may now leave if you wish.
Well done, everyone. All three of them were fantastic presentations. So thanks.
We're indeed.
Thanks, Kath.
Thanks,
Yeah.
Kath. That's awesome.
Yeah.
Right, good.
Thanks.
Yeah.
It was interesting to hear
Bye,
some more
everyone.
about what happened out Stony Creek Way, Brad.
Oh, one thing I have to say about Stony Creek, the gentleman, Luke from WA, said that creek, and I nearly jumped in and said, it's not that creek, it's only a couple of kilometres away from me where I'm sitting. It's our creek. Rightio.
Bye, everybody. See you later.
Thank you,
Yeah.
Drew. Thank you, Jay. Thank you, Brad and Richard and everybody, of course.